is PCR? PCR means polymerase chain reaction. So in this case, the polymerase chain reaction, it is a technique of synthesizing multiple copies of the desired gene, or we can say DNA, and this technique was developed by Academulus. In this technique, we have to take a sample of DNA, and by using PCR technique, we are able to make a multiple copies of DNA. So the basic requirements of PCR is DNA template, nucleotide primer, and the enzyme. So it carries out the temperature cycle. So here you can see, first of all, we will take a DNA template, that is target DNA, and the nucleotide primers and the polymerase enzyme, usually Taq polymerase enzyme for synthesizing of new DNA molecules. So this is the working mechanism of PCR. In this case, the three steps mainly occurs. This is how, as in cycle 1, denaturation, which occurs at 95 degrees Celsius. In this step, the DNA is heated to high temperature, heating resulting in the separation of two strands of DNA. And next, in annealing process, the formation of primer occur, which occur at the 55 to 65 degrees Celsius. And the last step is extension, which occurs at 72 degrees Celsius, which is the last and third step. So in this step, the enzyme DNA polymerase synthesizes a DNA segment between the primers, usually Taq DNA polymerase isolated from the thermophilic bacteria, they used in the most of the cases. So the two primers extend toward each other in order to copy the DNA segment typing between the two primers. You can see here there is an extension between the primers, which occurs very beautifully. So this step required the presence of deoxynucleotides, triphosphate, and magnesium ions. Again, cycle 1, 2, 3 occurs. Same here, denaturation, annealing, and extension. So during the 3 cycles, the copies of target DNA form 8 variable length DNA fragments. In cycle 4, so this is how we can use many cycles. With the help of this technique, we are able to make a billion time or many copies of desired DNA. So now this is the end of the topic. Thank you.